We are back in the Flagship Studio here at the JP Morgan Healthcare Conference. I am joined in the studio by Guillaume Pfefer, the CEO of Sale Biomedicines. And Guillaume has been on this stage a couple of times with us, so thank you for being back.
Absolutely. Thank you for inviting me back.
Great, great. So Sale was formed about a year ago, the combination of 3 companies, and you've had about 1 full year of operations, more or less. Tell us about the last year and the progress that you've made that has given you increasing confidence that this was the right combination of companies to bring together.
Yeah, what a nice question. I mean, it's first full years as one company coming from 3. We made that decision as a very bold move because we strongly believe on the power of RNA medicine and the potential that we can still reach with this technology across multiple diseases, not only vaccines. So the whole idea was to address the critical challenges and put 3 key components that we think bring us the ability to get RNA medicine to the next level. And RNA technology, we're going to talk about it maybe later, and absolutely novel the way to deliver RNA, and an AI company that also helps us to integrate the design of RNA medicine all together. When we talk about integrative AI generation, we integrate the design of RNA and the delivery of RNA at the same time. So 1 year in, I can now state that we had a start with a bang year as our first year as Sale Biomedicines. Our scientists made tremendous progress on both platform and the pipeline of 6 programs at the moment. We pick up the 6 programs to create the highest possible value for shareholders and patients, but also to be able to create a diversity of therapeutical area we can get into. I'm going to go super fast on each of them, but they're quite remarkable, and the level of performance achieved totally validates that the decision we made at that time of putting cells together was the right one. So let's start with autoimmune disease, with in vivo CAR. Okay. Here we can design nanoparticles that target T cells, RNA that express very potently CAR at the surface of T cells. All together, we achieve total and complete depletion of B cells in bone marrow, indicative of a reset of B cells, exactly what you want for autoimmune disease. We're looking at the first in vivo, off-the-shelf, short regimen, transient way to basically reset patients affected by B cells-driven autoimmune disease. GLP-1, a space where you will not necessarily expect mRNA to get into that space. Sub-Q dosing with a CAR that is set up to RNA, GLP-1 that is set up to fit the very narrow therapeutical window with duration. So we're looking at what could become a 1-month sub-Q dose, repeat dose of GLP-1 expressing RNA, which gives us the ability to bring the large-scale manufacturing capability of RNA, as we all know. We're also in rare disease, liver rare disease, where we've been able, in inborn error of metabolism disease, with one first disease that we're focusing on, to have a leverage the durability of expression that RNA bring versus mRNA. And we have likely a 1-month dosing here interval. Best benchmark is 2 weeks. So you see also here a very nice performance. We have 3 partnership, or 3 programming partnership, CFF with CFF and Pioneering Medicine on CFTR, where we had achieved extraordinary results that we reported at the beginning of last year. And also an announcement we made at JPN last year, working with the Gates Foundation on malaria. We decided to join the fight against malaria and decided to move into that space with 2 distinct programs, encoding of monoclonal antibody into RNA for prophylactic monoclonal antibodies, and also vaccines with an antigen encoding RNA. Here, the team at Johns Hopkins University will run the challenge studies on our AI-generated RNA. Found something quite remarkable. We outperformed the commercial product with regard to prevention of liver burden, and for the very first time in their own world, never seen before data as far as achieving sterile immunity with parasite level in the blood lower than detection level, which basically is what you need to stop this propagation of a disease that affects 250 million people in 2023 and killed 600,000 people in 2023. We're super excited about this progress. 1 year in, start with a bang, integration, putting these things together, the speed at which we've been able to bring that technology and outperform in this multiple disease area I'm actually quite excited. I think we made the right move.
Great.
And I cannot wait to see where we go from there.
Absolutely. Incredible year for you. So you touched on some of the characteristics that are inherent in the platform, the fusion of these three companies. And as a field, in biotech, a lot of us are familiar with RNA medicines.
Sure.
It's an area that is increasingly established. But you are bringing eRNA to the space, as well as that AI component, as well as the delivery component.
Yep.
How do those three components coming together help you overcome some of the challenges and the spaces that RNA can't go into?
Yeah, it's another great question. I mean, we're very uniquely positioned to address the challenges that our predecessors very well identified and unlock the full potential of RNA medicine. The time we formed Cells, we instantly created a new leader in the space of RNA medicine. We aggregated companies that were very mature. Some of these companies have more than 7 years already of life, from incubation in our flagship labs to where we were at the time we brought them together. So the progress is also a tribute of the maturity of this platform and the criticality of putting them together. So what are the two challenges that the field is facing? We saw the opportunity absolutely clear, but these two last remaining challenges prevent us to bring the full potential of RNA medicine. First one is pharmacology. The first generation linear RNA is not a stable molecule, which means that you send a very short-lived instruction to the cells to produce the protein. And that basically is not asking the cells to work hard enough, long enough to produce a long-lasting or a pharmacokinetically modulated protein expression. That limits where you can bring the current mRNA from with regard to disease area.
Yeah.
What we bring with the RNA is a much more stable message. The cells work much longer, harder, producing more protein over time, increasing the therapeutical power from the instruction we provide to the cell. The other thing is obviously the delivery, absolutely critical challenge. Very few cells we can recruit today with the current way we deliver RNA. And here in another company that we had in our portfolio, we have demonstrated that actually we can tap into what nature has created and harness a natural programming language to give us the ability to shape totally differently the chemistry at the surface of this nanoparticle with the eRNA in it, to bring the message in other type of cells, also making this nanoparticle immunoscience, which is what you want for frequent repeat dosing, including sub-Q.
Yeah.
The critical piece here is like better technology on the two fronts, but it's the coming together that brings the extra kick here. That's where AI come into play. And AI integrative generation of RNA medicine is actually a new concept, because we have now the ability to encode not only the delivery, but also the RNA, and all together with AI. The rapid progress you've seen, and that is unique in the industry. We're absolutely an outlier. We forgot what we bring to the table technologically speaking, but the stitching together makes us absolutely an outlier. The rapid progress you saw in 24 is already the demonstration. All of that bring performance above industry level in the first 6 programs, pick up for the diversity. So again, unique, very powerful value proposition from cell biomedicine.
So longer lasting, ability to deliver beyond the liver, and applications in a lot of different disease areas that have not been previously accessible using RNA.
Absolutely.
Yeah, that's really exciting. And what could be next in terms of 2025 for sale?
If 24 has been a start with a bang year, we would like 25 to be a prove it year. We've got to bring in value, right?
Great.
And tangible value. So we're going to be looking at nominating our first drug candidates this year. We're also going to continue to create abundance of data, including not only in rodent, but also non-human primates. So we're going to learn faster with more data. We're also going to learn in a way that translates in human by having access to higher species. And also, obviously, we're going to be looking at partnership as a critical way to build the company. We already secured 2 partnerships, and 3 programs are already in partnership. JPM and the discussion leading to JPM let me believe that we may have some good news in 25 with regard to partnership.
Terrific. So we've got some progress heading into the year. It's going to be a prove-it year for Sale. We look forward to welcoming you back here next year and see how you did.
I'll be delighted to be back.
Great, great. Thank you,
very
Guillaume.
much.